and welcome to today's webinar for NeuroScientific Biopharmaceuticals. My name is Nicola Gazzatti and I'm co-founder of investor relations consultancy Corporate Storytime. Today I'm joined by NeuroScientific Chairman and Interim CEO Paul Rennie, who will be providing us with an update following the release of the company's activities report for the December 2022 quarter. This will be followed by a Q&A session. If you do have any questions, please use the Q&A function at the bottom of your screen and I will put your questions to Paul at the conclusion of his presentation. So without any further ado, I'm pleased to hand over to Paul, who will present a NeuroScientific Biopharmaceuticals update.
Thank you, Nicola, and thank you for the introduction. I'd also like to thank everyone who's taken the time to participate in today's presentation. Next slide, please. As an ASX listed company, we're of course required to display our disclaimer. I won't go through that in any detail other than to mention its presence. Next slide, please. So many of you be aware of the board, the management of NSB, and I would just like to give you a little outline of the presentation today. So first of all, I would like to talk about some of the key highlights and developments during the December quarter, such as the filing of a new patent around Interim, also some of the executive changes, etc. And we'll cover that information, and then we'll really get into the meat of the discussion today, being the update on the resubmission to Ethics Committee for our commencement of a phase 1 clinical study investigating Interim. I would like at this point to acknowledge that there has been a very significant amount of high quality work being undertaken by the company. And I would like to specifically call and shout out to Dougal Thring, who's our Chief Operating Officer, for his really excellent amount of work that he's done, and also his colleague Simon Scott, who's our Director of Clinical Development. Both gentlemen have done an outstanding job in bringing the company forward. And of course, also Abby McNish, who's our Chief Financial Officer. You'll see that we've got a very good financial position, largely as a result of Abby's management of the company's finances. So a very big thank you to all of the management who've supported me greatly over the last few months. Next slide, please. As I mentioned, some of the updates from the December quarter. NeuroScientific filed a new patent for Interim, which was studied in combination with one of the leading drugs already registered for the treatment of multiple sclerosis, which is of course Teva's Copaxone. Copaxone came from research done at the Weizmann Institute in Israel and has been a leading therapy for people with multiple sclerosis for many years. Copaxone itself is registered for relapsing remitting MS and marketed in 50 plus countries. At its peak sales, it was over $1 billion US, so it was a blockbuster drug. It has come off patent and sales have now decreased down to under $1 billion due to generic competition. Two major generic manufacturers are Mylan and also Sandoz. And recently I was reading a report from an analyst saying that the Teva sales are going to come under further pressure as Mylan has just introduced a new pricing of their biosimilar Copaxone, which will see the price decrease from around about $5,500 per course of treatment per year down to about $500. So very significant reduction in price, expecting further pressure to come on the Copaxone sales for Teva. So what NSB did was that we showed in a proof of concept study that was done by a specialist neuronal research company in France that the combination of Interim and Copaxone resulted in a significant increase in the three major areas that are really referred to as disease modifying drug in MS. That is seeing an increase in the neurite length, the neuronal cell survival or the brain cells surviving, and new myelin formation around the axon. So they are the key indicators. And what we found was that it was only the combination of Copaxone and Interim that resulted in a meaningful benefit in all of those areas of measurement, new neurite length, neuronal cell survival, and new myelin formation. Independently, EmptinB and Copaxone had an effect on those areas, but the largest and the most consistent effect was seen by the combination. So given that result, NSB has reached out to its contacts at Teva and provided them with some information which is being evaluated at the moment. But I think it's an excellent partnering opportunity for Teva or one of the generic companies who want to increase their presence in the MS space. Next slide, please. Also, during the quarter, we had some executive changes, and just providing you with an update here. As many would know that I'd stepped into the interim CEO role. I was very honoured to be offered the position, and it's been really a pleasure to be able to work with professionals such as Dougal and Simon and Abby. It's been a wonderful experience. I would also like to acknowledge that we've had very good support from some external providers, Dr. Jürgen Linder, who's been working very closely with our team on regulatory matters and also on the formulation. Jürgen is a person who has extensive experience in pharmaceutical formulations, and also Dr. Barry Levine, who is a preclinical toxicologist, US board certified. He has provided us with a lot of very important information as we've gone over the last few months. So thanks to all of the people involved. Our search for our full-time CEO is ongoing. We have identified some excellent candidates. We are at the short list stage and about to commence our reference checking on those candidates, but that is looking very positive for an appointment in the near future. And also Dr. Anton Yurov, he moved from executive director to non-executive director during the period of reporting. Next slide, please. Also, it's important, I think, for shareholders to understand that we've received approval for an advanced overseas finding in relation to an R&D tax incentive for our preclinical efficacy study focused on multiple sclerosis. So this is where the company asks the R&D tax office for a ruling to say, will this research that's being done, it's not being done in Australia, it's being done overseas because the expertise is not available in Australia, will that be applicable for an R&D tax rebate? And we got that ruling, and so therefore we do know that we have up to 1.62 million for costs associated with our preclinical MS MRI efficacy study. And that's obviously great news for shareholders. That's non-dilutive funding. Also, we have in vitro models which are being undertaken as well, which are also part of that 1.62 million. These are animal models of MS being used in association with novel MRI imaging techniques. So this is looking at the effect of the drug specifically on those areas of the brain cell, the axon, etc. And the results will potentially supply supportive evidence of EmptinB's disease-modifying effect in multiple sclerosis. Next slide, please. Okay, so that was the background to some of the developments that occurred in the last reporting period. But I know a lot of people, a lot of investors want to know what's happening with the resubmission for our phase 1 clinical trial. So just to remind everybody, people who've been following the story will understand that we received feedback from the failed submission from the ethics committee that they had 3 areas of concern, and I'm going to address each of those now. One was safety, one was the purity, and one was the efficacy. So the safety referred to some observations in the animal studies where some of the animals were vocalizing or in the case of dogs, yelping or whining, barking, etc., when the drug was administered, and there was some residual redness around the injection site. And the ethics committee referred to this as a safety issue. Now, we also did long-term systemic investigation in the animals to look at the effect of the drug in all of the major organs. And this is where Barry Levine has been very pivotal in understanding that from his perspective, there is no obvious safety issues or toxicity issues with EmptinB in a in-life model, such as this case within the dog. So we're not talking about major safety events. We're talking about this administration of the drug and a local effect around the injection site. Barrier identified was probably in relation to the formulation that was being used at the clinical trial centre doing the research for us. So we engaged with Dr. Jurgen, who then worked with our team to really start to evolve a formulation that was much more acceptable for a subcutaneous administration. So it had a different pH, it had different osmolality, and all of those factors needed to be checked and double checked to make sure that it's all in line with pharmacopoeial standards. So that research has been done, and we have got to a formulation now that we feel much more comfortable with. And we're now embarking on a bridging toxicology study, which is designed and has already commenced in terms of booking spaces with the CRO. So this work is about to commence, and this will assist us with confirming the local and the systemic tolerability of the new formulation of Emptin-B and its appropriateness for a phase 1 clinical study. So I should say at the outset, those three components of safety, purity, and efficacy that were reported by the ethics committee, all of the work that was identified as being perhaps a gap in our data set, all of the work that has been identified by the ethics submission has either been completed or is work in progress. So we have addressed all of the areas of concern, and we are in the process of filling that with new information to demonstrate to the level required by the ethics committee or regulatory authority that we have all the data necessary for us to go forward into humans. Next slide, please. So the next question was the purity of Emptin-B. So there was a question raised around the purity of the final drug substance. And we know that peptides are complex, and particularly when we're talking about a dendrimer being 4 strands of peptides, there can be inadequate bonding and breakdown of some of the peptides to amino acids, and some shorter peptides formed. And these are what we call related impurities. So we have asked our manufacturer in Switzerland to do an impurity identification on all of the available batches that we have. So these are some of the batches we used in our toxicology studies. These are some of the batches that we have had manufactured to GMP standard for use in humans. Go through all of those batches of available product and make sure that we can identify the impurities of each of those products. And I'm pleased to report that those impurities are related to the peptide itself. There's not any evidence of organic chemicals as impurities in the product. So we believe that we will be able to show that information to the ethics submission next time, to be able to show that we have very good understanding of what the drug substance is and what those impurities are. And those impurities of amino acids are generally recognized as safe. So we think that that's a very valuable piece of information that has been obtained. We're also very close to completion of a position paper asking the EMA, one of their regulatory officers, to provide a scientific advice to the company for our submission. So get the EMA to look at our submission and say, here's our proposed protocol, here's our preclinical, our safety data, et cetera, and advise us if they believe that the purity of the product as it's presented with this extra information of the identification of the impurities would be sufficient for a human clinical trial. And we think that the EMA would be an excellent regulatory authority to ask that question. And if we get a positive response, it opens up the possibility, if we have any further issues with regulatory submissions in Australia, we could always go to Europe and commence our phase 1 study in Europe, should that need arise. We've also put our GMP batches under a formal long-term stability program. So those stability studies are underway. They are all compliant with GMP regulations. And what we're looking at there is to make sure that when we come to subsequent trials, such as phase 2 and phase 3, we have all the necessary stability study data on file. So that's an extra piece of information that we're doing to help us with our submission, but also for future submissions as well. Next slide, please. Efficacy. So this is, again, where ethics made note of the fact that although we were talking about a phase 1 study going into humans looking at the safety and tolerability of Emptin B, our proposed future studies was going to be a phase 2 study in multiple sclerosis. And so they raised the question, well, you've submitted data on Alzheimer's, how does that relate, or how is that connected to a study in multiple sclerosis? What is the efficacy signal that you have for multiple sclerosis? So we took that information and feedback on board. We did then set up a study with an external CRO, and they are currently, or they have been working now for several months on a mouse EAE model of multiple sclerosis. We have seen some of the initial clinical outcome data from that particular study, which we have had very encouraging clinical scores. However, the issue that we're really looking for is about to come in a final report, which will be at the end of February, early March, which will look at the immunohistochemistry, so the staining of certain biomarkers within the neural network, looking at ensuring that there is some evidence of remyelination around the axons. So therefore, we have evidence to support the notion of moving into a phase 2 study at some future stage and having efficacy around Emptin B and its ability to remyelinate the axons. So that's a study that has been going on for a few months. And that study is, in addition to that, one of the questions from ethics was, how do we know that Emptin B crosses the blood-brain barrier? We have undertaken a second study, which is looking at Emptin B and its crossing the blood-brain barrier and getting that necessary data for the answering of that question. So all of the questions that were raised or posed by this ethics committee have been undertaken. All of that data is being generated. So we believe that we're going to be in a very good position, and we hope to make the resubmission around the middle of this calendar year, taking into account we still need to get some information from the bridging tox study. We also need to get the final report about the remyelination study and compile all of that and put it into a comprehensive package of information, which will hopefully then go forward and go through the ethics committee and get approved this next time, given the fact that we're addressing all of the issues that were raised in the last submission. So we're looking to bring all of that data together and compile it into a document and then resubmit around the middle of this calendar year 2023. I can't be any more specific than that because we do wait on the final reports from the external providers, and they give us a window of, it could be this month or next month, without actually narrowing it down to a particular day. And of course, in preparing these documents, we have to make sure that they are quality controlled, and we make it very, very clear that the inadequacy of the first submission has been addressed with this data, and we have to make it a very coherent story that all the data points to this being a compound that is demonstrated to be safe and has evidence of efficacy within the animal models that have been chosen. Next slide, please. Now, just touching on the financials, one of our last updates, we spoke about the fact that we believe that we had sufficient cash to get us through this bridging period of where we're getting all of the necessary data to go back to ethics and pay for all of the necessary studies that were required to bridge the gaps in our data from the last submission. And we felt that we would have enough cash to get to that point and make the resubmission. And at that point, we would then be able to disclose our final cash position. So it's again a recognition of the great work that Abby and her finance team have been able to do, that we had a cash position of $3.3 million at the end of December 2022. We continue to maintain a strong cash position by making sure that we have a very tight control over our expenditure. We also, we've identified here on the slide some of the operating cash outflows for the December quarter, but also I want to make mention that we did recently receive $3.74 million R&D tax refund. So that will help our cash position, and again confirm that we should have sufficient cash to keep the operations going, to get all the necessary data, to address all of the gaps in the first submission, and make a resubmission and hopefully have it successful and come out the other side with a phase 1 clinical study that has been approved by ethics. Next slide, please. Okay, so just a reminder for everyone, I think over the journey, we have been able to demonstrate very clearly that MTB has generated enough signals, whether they be in vitro models or in vivo models, that MTB has all of the hallmarks and signals of being a truly disease-modifying drug, particularly in the area of multiple sclerosis. So we're talking about it does stimulate and preserve the brain cells or the neurons. It does promote the axon regeneration and the myelin around the axon. It does address neuroinflammation and downregulate the inflammatory responses in the brain. And also we know from the work done by our group in France that MTB does promote remyelination. So that's the end of the presentation and the update, and I'll now hand over to Nicola to address any questions that might have come from the presentation.
Thank you so much for that update there, Paul. It was fantastic. So we do have a couple of questions, but we would like to acknowledge some that have come through in relation to the that were sent in advance and regarding the status of the resubmission of the Human Research Ethics Committee approval for phase 1 clinical trials for MTB. I feel you've probably covered the majority of that question in your presentation. So we will move on to our next question, which is, in the event that MTB is sold to Big Pharma, will NeuroScientific be looking to add to its drug pipeline by acquiring other compounds or assets for development?
Thank you very much, Nicola, for the question. And yes, I can say that NSB is actively engaged in the evaluation of a number of new projects that will lead to potentially clinical programs in the future. It's obviously far too early in the process to disclose all of those, but we do have a diligence committee that is working on the evaluation of a number of projects. We do recognise that should we be in the fortunate position of a partnering deal with a pharma company, we do want to continue on the development of projects in the neuroregenerative space, and therefore we will look to build out our pipeline over time. But it's not something that is easily done, and it's not something that is done quickly. So we have to work our way through it, and we expect that the new CEO will also have some ideas around what would be good opportunities for new products going forward, and that will be one of his or her key foci for the future, which will be building out some new projects.
Okay, thank you, Paul. So our next question is the company looking to add board members with technical and scientific experience?
Yes, I think, well, the plan is, of course, that once the full-time CEO is appointed, I will relinquish my interim CEO role and go back to a non-executive chairman. We do have Dr. Anton Yurov on our board at the moment. But as time goes by, yes, I think we will start to develop further scientific and clinical experience on the board. But I think at the moment we have a very good board, and particularly acknowledging Stephen Quantrill. Stephen is not a scientist, but Stephen does bring very comprehensive and solid business advice to the business, which of course is very important, as well as the scientific and medical aspect of the company. So we need to make sure that we always have a good blend between business acumen and scientific and medical acumen. But I think the events over the last year or two have probably identified to us that we need more expertise in the regulatory and the clinical development side of the business. And it's no easy task. I mean, there are many companies who really have great technologies, but getting to a clinical development program takes a lot of time and resources and expertise, and that's something that we want to do. So to answer the question, yes, we will be adding further skills to the board over time, but it won't happen next month. It will happen over a period of the next 1 to 2 years.
That appears to cover the majority of the questions that we've received from our audience today. Thanks to Paul for his time today, and thanks to the NeuroScientific team for all their work. And thanks again, Paul.
Thank you very much, Nicola. And could I also just thank everyone who's tuned in and remind them that if they do have any further questions, we are always available. My email address is on the last slide of the presentation deck. If people want to get in touch, send an email or make a phone call. More than happy to talk through any questions the investors may have. So thank you very much, Nicola. Thank you, Lucas. And thanks again to the NeuroScientific team and everyone who's attended.